I want to welcome D.A. Gross, who is the CEO of Eledon Pharmaceuticals. And would you mind, D.A., introducing yourself? And for the people who haven't heard of Eledon before, can you tell us a little bit about your company in regards to the three big pillars that you're working on, type 1 diabetes, kidney transplant, and xenotransplant programs, and just kind of give us a broad overview of what you're working to accomplish?
Great. Thank you, Devin, and thank you for having me on. I am the CEO of Eledon. I co-founded what is today Eledon with our president and chief scientific officer, Steve Perrin, about 5 years ago, almost to the day. We are a company that is principally focused on transplantation medicine. Our roots are also in ALS. Tegopubart, which is our lead asset, was initially discovered at the ALS Therapy Development Institute. And when we in-licensed or acquired the drug to create what is now Eledon, we expanded the focus to be on transplantation as well. So in transplantation, our three primary areas of focus are islet cell transplantation for type 1 diabetes, which we'll discuss today, kidney transplantation, so to protect transplanted kidneys after they've been transplanted in patients receiving kidneys for the first time, and then xenotransplantation, which is putting organs from other species, in this case from pigs, into humans. And we've as we'll discuss, I'm sure, we've now done that a number of times, including with hearts and kidneys. And finally, we continue to believe in our ALS program, although right now that is on hold, principally for financial reasons as we look for ways to move it forward.
Great. Thank you for that. I appreciate it. So now that everybody has kind of a broad overview of your company, can you walk us through how Eledon first got involved in the type 1 diabetes space? And can you kind of share with us what your mission is in regards to type 1 diabetes?
So we have been involved in the type 1 diabetes space since the first day that Eledon was created as a company. So when today's Eledon was formed, about, as I mentioned, 5 years ago, we brought in Tegopubart that beforehand had been developed in ALS, and we expanded the focus in transplant. And that since that time, the initial transplant focus was in diabetes. And so our original transplant experience was using a non-human primates or monkey version of T1D, and using islet cell transplantation to see if we could get to a functional cure in those animals. And when we founded Eledon, that was one of the key areas that we were focusing on, and that our investors decided to fund the company to pursue. So our focus on type 1 diabetes actually predates our focus on kidney transplantation or xenotransplantation.
So for the last 25, 30 years, tacrolimus has been the standard immunosuppressant in islet cell transplantation, but many clinicians and patients recognize that it comes with significant challenges. And as you've mentioned, those include beta cell toxicity. These cells that it's supposed to protect, it's actually toxic to these cells, which is crazy. You can increase the infection risk. There's long-term effects like being toxic to the kidney and nervous system, as we discussed, and terrible tremors. These tremors are debilitating for many people, as well as leading to hypertension and insulin resistance over time that does require insulin replacement. So having said all that, could you walk us through why you think Tegopubart might have a safer and more effective alternative for people with type 1 diabetes based on the data you've seen so far?
Sure. So when we founded Eledon and brought Tegopubart forward or into transplantation, our goal for allotransplantation, so that's for transplant between a human to another human, was to replace tacrolimus. And the reason why we wanted to replace tacrolimus was multiple-fold. The first one has to do with the side effect profile of tacrolimus, which you've just mentioned. Tacrolimus, and calcineurin inhibitors more broadly, while they helped open modern transplantation medicine and make it possible, come with a wide range of side effects. In particular, they are toxic to the beta cells of the pancreas that make insulin. So in patients that, for example, get kidney transplant, it's very common to see both high blood sugars initially, as well as ultimately something called post-transplant new onset diabetes. So this is insulin-dependent diabetes that occurs because tacrolimus has destroyed so many cells in the pancreas that patients are no longer able to make sufficient insulin to be able to control their blood sugars. Beyond the effects on the pancreatic cells, tacrolimus is also directly toxic to the kidneys. Now, if we look at patients that have, for example, heart transplant, about 1 in 11 over time will need either dialysis or a kidney transplant as well. And the reason those patients that have received a heart transplant end up having kidneys that fail is not because of their underlying kidney disease. It's because of the tacrolimus that over time can destroy their native, originally healthy kidneys. Tacrolimus is also associated with causing high blood pressure, as well as some symptoms that patients particularly dislike, which are tremors and brain fog. Now, I want to highlight it continues to be an amazing drug, one that has helped make modern transplant medicine possible, but we believe that there might be more novel approaches to do that that could not only be more effective, but could also be safer. And so it's with that in mind that we brought forward Tegopubart. Tegopubart is a very targeted agent. It interacts where the T cells, which are a type of white blood cell that are almost the police officers, if you will, of the immune system, it interacts with how these cells communicate with other parts of the immune system. And so by changing how that communication is occurring, we inhibit the immune system from being activated and being able to attack the transplanted cells or organs. And we can do that in a way that doesn't have the what's called off-target effects, so acting in other places where one may not want to see impact, and in particular on the effect on the kidneys or islets or blood pressure or the brain or the nervous system. So to date, a lot of the side effects that we've just described that are traditionally seen with tacrolimus and calcineurin inhibitors, we're not seeing with Tegopubart because we're a more targeted therapeutic that doesn't bind to the places that are causing issues for the older drug.
Gotcha. Thank you for that explanation. Can you share with us the most recent, up-to-date, where we're at currently with the breakthroughs of your type 1 diabetes program, including how many patients in your trial have become completely insulin independent?
Sure. So we are currently running, or the University of Chicago is running, and we're supporting. It's an investigator-sponsored study, and it's being funded thanks to some grants, in particular from Breakthrough T1D as well as the Cure Alliance, a study in patients with type 1 diabetes that receive a islet cell transplant, and for immunosuppression, so to protect the cells from the immune system, so that the immune system doesn't immediately attack and try to destroy the cells, we're using a number of immunosuppressants, but the cornerstone is Tegopubart. And in this study, Tegopubart is being used instead of the traditional immunosuppressant, which is tacrolimus. The study is funded for up to 9 or 12 patients. To date, there have been 6 patients enrolled, and so that have received an islet cell transplant and are receiving Tegopubart as the cornerstone of their immunosuppressive regimen. And we've publicly presented on the first 3 subjects, the first 3 patients that were transplanted. And what was exciting was that all 3 of the patients were able to get off insulin, and so they're all insulin-free today. But we saw the cells rebound and begin to function very quickly. So we saw a rate of what's called engraftment. So it's the cells being able to take the position that one wants them to take in the liver and to begin functioning. That was very high, higher than what has historically been demonstrated with tacrolimus. And associated with that, we saw nice blood glucose control that started very early, within the first few days, the first 48 or 72 hours, in fact, after the transplant. And from a side effect perspective, as I mentioned, and as you brought up, one typically sees a number of side effects when patients are on tacrolimus, and to date, we have not seen those side effects. Not only in islet cell transplantation, we actually haven't seen those side effects, for example, in our kidney transplantation study that's ongoing. So it appears that we can protect transplanted cells or transplanted organs, and that we may be able to do so without many of the traditional side effects seen with tacrolimus.
That's incredible that you're already seeing blood glucose control within 48 to 72 hours, as you said. That typically doesn't happen with tacrolimus nearly that early, does it?
It can take longer to see the blood glucose control. Now, to be clear, early on, the patients are still requiring exogenous insulin. So they still need, after the transplant, to take insulin as the new cells kick in and begin to make insulin. But we're able to see quick reductions in the amount of insulin that patients need, so that they can see very quickly a reduction of their needs, typically within weeks. And then we've even had a patient, one of the 3 patients was able to get off insulin, I believe, within the first month or so. So it can be quite fast.
That's incredible. 1 month. Do you know the average time for tacrolimus patients to become completely insulin independent?
My understanding is that it's longer than that. It's that it can take a longer amount of time. What Peter Witkowski has presented, who's the physician that's overseeing the study, is that on day 75, he's seeing more engraftment, so more cells that are functioning when patients are on Tegopubart than when they're on tacrolimus.
That's incredible. It's incredible what these cells can do when their mitochondrial function isn't being compromised by a CNI, right?
Right now it continues to be a small end, but we're obviously excited and hopeful.
Thank you. Studies have shown us that only about 10 to 15% of patients that receive an islet cell transplant with tacrolimus are able to remain insulin independent at the 10-year mark down the road. So based on the data you've seen so far, how confident are you that Tegopubart could improve the long-term durability of insulin independence? And what are the key factors that might drive those outcomes?
Sure. So with us, to date, we are still in the early days of testing Tegopubart, and that's true both with islet cell transplant as well as with kidney transplantation.
Right.
But if we look at our experience with both of them to date, and to date we've now had over 100 patients that have had transplants where they've used Tegopubart as the cornerstone immunosuppression. What we've seen is that whether it's islet cell function or kidney function, it tends to be quite good and better than what one would expect to see if those patients had been on tacrolimus. And because tacrolimus has a negative impact on both kidneys and islet cells, one would expect, and indeed one sees, that the longer patients are on tacrolimus, they see a negative impact on both their kidney function as well as their islet cell function. Now, since Tegopubart was designed not to have that negative impact, and as I mentioned, we're not seeing a negative impact on kidney function or on islet cell function tied to Tegopubart, one would expect, and we believe that over the long term we'll be able to show better function and better graft survival over time. So what we want to do is to help increase how long transplanted cells and organs survive, so that we could see more patients remain insulin free going out to 10 years, and so that we may also push back the amount of time that kidneys are working, so increase the amount of time that kidneys are working, so that patients will need fewer repeat kidney transplantations as well. So our goal and our expectation is that if Tegopubart works the way we believe it can and it will, transplanted cells or organs will be able to function longer than what they do today.
Thank you. I'm very optimistic as well about the long-term durability of your drug in protecting these organs based on the data that has been presented so far. So looking ahead, can you give us a sense of what's next for the type 1 diabetes trial, including how many patients you expect to enroll and the key milestones or timelines you're aiming for? I know you mentioned that there's been an allocation for 12 total patients in Chicago with Dr. Witkowski to be dosed. Is there anything else you can tell us about what lies ahead in this trial?
Sure. So in islet cell transplantation, we actually will have 2 studies that should be enrolling in the near future. The first study is the investigator-sponsored trial from the University of Chicago that we've been talking about. That is now going to be a 12-person study. We have 6 patients enrolled today. We expect to have 9 patients enrolled by the end of the year, and so 12 going into next year. In parallel to that, there is another study launching, another investigator-sponsored study. This one is going to be at multiple centers, including, of course, the University of Chicago, and that will be for patients that have both type 1 diabetes as well as kidney malfunction. And so these patients that have both T1D with kidney disease would be eligible to participate in that trial. And this is a population where doing an islet cell transplant would be problematic because the tacrolimus would obviously have a negative impact on these already diseased kidneys. So the hope here is that we may be able to do islet cell transplants and help relieve type 1 diabetes while at the same time protecting the remaining kidney function. And so we expect that study to be active and recruiting next year. Overall, what that means from a time to market, if you will, or an ability for us to help make this available to as many patients as we can by having the drug approved, is the following. Our goal is, once we have about 9-10 patients worth of experience, which should be pretty soon, is to then go and talk to the FDA about what a path to approval could look like for Tegopubart in islet cell transplantation. And so since we expect to have 9 patients in by the end of the year, that means that next year we expect to be in a position where we can talk to the agency and see what the agency would like us to do, what type of trial or trials the agency would like us to run, so that we may one day get Tegopubart approved for islet cell transplantation in the United States.
Thank you for that. Is that is there a number of patients that the FDA typically likes to see data from before they would grant approval, based on other trials from other companies?
So that's a great question. Every trial is different, every company is different, so it's hard to extrapolate what that might look like. There is one company that had an approval in islet cell transplantation, and that's a company called Celltrans that had its islet cells approved. And of course, there's Vertex that has a path to approval for its islet cells. And in the case of both of those companies, it's in the 40 to 50 patient range that the FDA had asked. Now, those are for cells. We're obviously a therapeutic, so it's not quite apples to apples. But if one were to look at a comparator, that's probably the most natural comparator to look at. We're at the cutting edge here. So it's going to be us approaching the agency in terms of now a new therapeutic to go along with cells that will be leading this type of a discussion.
Thank you. For my own information, my daughter being a 6-year-old patient with type 1 diabetes, if there was approval granted down the road for Tegopubart for the use of islet cell transplant, is there a potential? How does it work exactly? It's going to be 18 to 65 first, and then can they potentially peel back the age as they observe the safety and efficacy profile performing very well? Could they then peel back the age to maybe treat patients of a younger age? How does that process work? Can you speak to that?
Sure. So our goal as a company is to replace tacrolimus across every single type of transplantation, regardless of the source of an organ, so whether it's coming from another human, whether it's coming from a pig or a xeno, right, another animal, and whether that human that donated the organs or the cells was a living donor, like a sibling or family member, or a deceased donor. And regardless if it's cells, whether the cells came from a donor or were man-made. And so what we want is to try to give anyone who might have a benefit from Tegopubart the opportunity to benefit from Tegopubart. And that would, of course, include both adults as well as children. So you are correct that our early studies in kidney, in islet cell, as well as in xeno have been in adults. And that has to do with the way drugs are typically developed. We start with adults because the safety bar is higher, often with children. So we're starting with adults as well. But our plan is absolutely to expand the use and to do whatever trials necessary so that younger patients, children, may get access to Tegopubart as well. And that's going to be especially true, by the way, in xeno, since there are certain types of transplantation, particularly heart transplantation, where one of the biggest needs is in kids and infants. And we've presented data in infant cardiac xeno. This wasn't done in humans, but in non-human primates, in baboons. and there, as an example, the primary focus would be specifically for children, to help children. So as we look forward, our goal is to help advance the science, to help advance the potential of Tego for anyone who might need it. And so that will definitely include children if we can, so that people like your daughter will hopefully be able to benefit one day.
Thank you. How does the FDA typically handle these approvals? Would they peel back to like 16 or 14 or 12? Would they go 12 to 18 and do a separate trial on that age group?
Exactly. So typically, as one looks at younger patients, one needs to do more trials. So we will work with the agency in order to do whatever studies necessary, so that younger individuals may be able to hopefully benefit from Tego, and we can make sure that even in children, right, growing children, that Tegopubart is safe and effective enough for use at their relative ages.
Thank you.
But we would expect to need studies for younger individuals as well. And that is our plan, and that's our development plan.
Awesome. This is a little off topic, but we talked a little bit about the monkey trial earlier, and if I remember correctly, some of the monkeys continued to show insulin independence after the discontinuation of the trial drug. Is that a potential? Do you see that potentially happening with humans with Tegopubart?
So when we created Eledon, our first focus area in transplant was islet cell transplantation. So we worked on islet cell transplantation, in fact, before we worked on kidney transplantation. At the time, we had done work with Norma Kenyon at the University of Miami, and in animals and monkeys that had that had the equivalent of type 1 diabetes, what she showed is that if she did an islet cell transplantation and she used Tegopubart instead of tacrolimus, the monkeys tended to do better. And what I mean by that is the following. The monkeys were growing more, and they had better control of their blood sugars. So they made more insulin and were able to have better insulin control than equivalent monkeys that were being treated with tacrolimus to protect those cells. And these were monkeys that were being treated with only one drug, so monotherapy. So it becomes more apples to apples, because it's really how did the monkeys do when they were receiving tacrolimus versus how did those monkeys do, or different, not the same monkeys, right, but different sets of monkeys do when they were receiving Tegopubart. In terms of what you mentioned, which is tolerance induction, or the ability of people or animals to continue to see protection of transplanted organs even without immunosuppression, we saw something striking in those early studies with Dr. Kenyon, and was the following. Maybe to take a step back, we should remember that in transplant, patients that receive a transplanted organ or transplanted cells need immunosuppression for the life of their transplant. And that's because our immune systems are set up to find things that are not self, that are foreign, and to attack what's foreign. And so in the case of an infection, the immune system will attack a virus or a bacteria because it's foreign. And that makes sense, and that's good for the organism. Of course, in terms of transplant, it's the opposite. What we're trying to do is to prevent the body, the immune system, from attacking and destroying the organs or the cells, because we want that organ or those cells to survive and function. Now, the immune system continues to be active and vigilant throughout one's life. So patients that receive a transplant need to be on immunosuppression as long as they have that transplant. If a patient stops taking their immunosuppressant, then that patient's immune system will immediately reawaken, see the transplanted organ, and begin to attack it. And that's true not only in humans, but in animals as well. So, with that as background, what was striking in the work done by Dr. Kenyon was that she saw that in animals that had been on Tegopubart for a year, when she took them off therapy, so when she stopped giving them Tegopubart at the end of the year, the animals continued to have cells that functioned and were not seeing rejection for up to 6 months. So these animals had no more drug on board because Tegopubart only has a 3 or 4 week half-life, and yet they were continuing to see the effect. And the belief is that what occurred was a reprogramming of some of the immune cells to turn them into protective cells that would then protect the organ. So earlier, when we spoke about what the mechanism is for Tegopubart, I mentioned that Tegopubart acts on the police officers of the immune system, the T cells, to change how they communicate with other cells in the immune system. And it ends up that these T cells are not only cells that say what to attack, but there are a subset of them that tell the immune system what to not attack. They're called T regulatory cells. And so we believe that when we interfere with how T cells communicate, a subset of those cells will end up becoming T regulatory cells and have a protective effect as a result on the transplanted organ. And in fact, recently in August, we presented data on liver transplantation in monkeys, so non-human primates. And once again, we observed something very similar, where when animals were taken off anti-CD40 ligand at the end of a year, the immune system, instead of immediately reactivating the way one sees with tacrolimus, the immune system continued to allow, in this case, the livers to function without an immune attack for months on end. So what would this mean long term for patients? It may mean that in a long time, there could be a way to get to the holy grail of transplantation, which is no immunosuppressant needed. And to that end, we are working with Harvard Mass General Hospital, where we expect to launch a tolerance induction study using Tegopubart. And so this is in kidney transplantation, but it would be a study to begin to study to see, is there a way that using various approaches, including the use of Tegopubart, one could get to a state where after a certain amount of immunosuppression, patients could be off all immunosuppressants for a very long time, ideally for life. So this is really the cutting edge. I think what we're working on more generally is cutting edge. Now this is moving right to cutting edge twice removed, if you will. But the reason we're working on that is based on the data that we saw initially in islet cell transplantation in the non-human primates, and more recently in liver transplantation, again in non-human primates.
Thank you for that explanation. That's fascinating. It seems like the Tegopubart can more or less train the immune system with these Tregs, and you may not need to continue taking Tegopubart potentially long term.
I think that's so. The ability for tolerance induction is that's very different from and is many years away. To be
Right.
clear, if that might be possible. But the belief is that in the nearer term, taking Tegopubart should lead to better outcomes because of better efficacy, the potential for better efficacy, as well as the potential for better safety. And here, the efficacy and safety are very much tied. So in islet cell transplant, part of the safety issues with the current standard of care with tacrolimus is its negative effect on the beta cells of the pancreas. So if we're not seeing those negative effects, that has both a safety advantage as well as an efficacy advantage in islet cell transplant, since more of the resulting cells should survive and function better.
Thank you. So Eledon is addressing significant unmet needs from type 1 diabetes, kidney transplant, and xenotransplant. Looking ahead, how do you see the market opportunity for Tegopubart across these three different areas?
So we think there's an opportunity to help many patients in all three of these transplant areas, as well as other types of organ and cellular transplant as well. We discussed a lot islet cell transplantation today. As we look at the future of islet cell transplantation, our goal is to be agnostic to the type of cells being used. So we are living an evolution in what's possible in islet cell transplantation in terms of where the cells are coming from. Today and traditionally, all the cells have been coming from deceased donors, and that's where, and that's the experience that we have to date. We're seeing the market evolve with man-made stem cell derived islets, as well as potentially xeno-derived islets. So islet cells that would be taken out of animals, out of pigs. And there's some very exciting data that's come out of China as an example. So for us, what our goal is to be able to help as many patients as possible if we can. And so we are active across all three of those areas when it comes to islet cell transplantation. In terms of kidney transplantation, today we're focused on patients receiving kidneys for the first time. So they're the so-called de novo patients. But over time, we'd like to be able to help patients that have already received one kidney and might need another kidney, or might be experiencing side effects with their current standard of care, with probably their tacrolimus, and allow them to switch onto Tegopubart. And that would be different studies. And then finally, for xenotransplantation, our partner in kidney transplantation, eGenesis, has now received a path to approval from the FDA. So there's now a path to potentially having them get approved with their porcine, so their pig kidneys. And we would look to continue to work with them there, but we also work with them on other types of organ transplantations. So our goal again there is to be active and to be available wherever we can. Overall, the transplant market is one that has been hampered by not having enough organs. We all hear about the organ shortage. In kidney transplant, for every 5 or 6 patients that we transplant in the United States every year, one person dies waiting for an organ that they don't get. So one of the hopes is that using Tegopubart can help with the organ shortage as well as the islet cell shortage that would occur to have sufficient islets. And the way we would do that is in islet cell by developing Tegopubart so that it may be used in conjunction with any islets, as I mentioned, human donor islets, man-made islets, or xeno islets. And then more broadly for other organs, we believe we can help alleviate the organ shortage uh, needs today in two ways. One, by allowing organs to survive longer. So if organs survive longer, then there would be less need for repeat transplants. And those organs that would not be used for a repeat transplant would then be freed to go to someone that otherwise would not have received an organ. And the second way is, of course, xeno, which could help completely resolve the organ shortage, since we could manufacture organs, if you will, in pigs.
Thank you. Going back to the stem cell based islet cells, can you see a potential collaboration between Eledon and a company that can produce those cells to open up an unlimited supply of cells for patients with type 1 diabetes in the future, potentially?
So we collaborate actively today. Obviously, in xenotransplantation, as we mentioned, we're collaborating closely with eGenesis in the kidney space, as well as other organs. But the goal looking ahead is to work with other companies that are moving forward organs and cells, and that's whether they're man-made, so islet cells that are stem cell derived, or that could be produced through xeno. So we would absolutely look to collaborate with others in order to try to make Tegopubart and ultimately to help accessible as widely as possible, and ultimately to hopefully help as many patients as we can.
Wonderful. So as we wrap up, is there any message that you'd like to share with the type 1 diabetes community that have been following your research closely? I have many followers that have been hanging on every update that you release, and we love to hear everything. Is there anything you want to specifically say to the type 1 diabetes community as we closely watch your very exciting work and trials going on for type 1 diabetes?
We are dedicated to type 1 diabetes and to helping in whatever way we can. As I mentioned, type 1 diabetes is in the DNA of this company. When we founded Eledon, the original data that we generated in transplantation was in T1D. And the first transplantation study that we launched was an islet cell transplant study. So this has been at the core of our focus from the beginning, and will very much remain at the core of our focus moving forwards if we can. The other aspect is tied to how we created the company. This is, Eledon is not a company that came out of big funds, big venture capital creating the company. This is a company that was initially created coming out of the nonprofit world. Tegopubart was developed at the ALS Therapy Development Institute, a nonprofit focused on ALS. And the ethos that comes from being affiliated with a mission-focused nonprofit continues to permeate everything that we do today in terms of how we think about working with and existing for patients. So it's been at the core of who we are, at the core of how we make our decisions, and it will continue to be at that core in the future as well.
Thank you. And I just had one last question to throw in.
Sure.
You mentioned once you get 9 to 12 patients data for type 1 diabetes, you can then go to the FDA and have them start taking a close look at it and getting feedback from them. Do you have any timeline of what could potentially an approval look like for Tegopubart as a therapy for islet cell transplant? Just as far as people, there's been the joke that we're 5 years away from a cure for 50 plus years. What are we talking a couple of years, of 5 years from now? Any estimate on timeline?
So I completely hear you. Let me start in terms of the joke. If we're five, right? We've been 5 years away for 30 years, or 5 years away for 50 years.
Yeah,
That might have been true, but we're in a different place today from a scientific perspective, and we can all sense it, right? We're all living at a time when there is a transformation going on in
absolutely.
in transplantation. I mean, you see it in, if we go look at xeno for a second, because we've been 5 years away from xenotransplantation for 30 or 50 years as well.
Yeah,
But today in xeno, there is a gentleman who is living today with a pig kidney, who's had that kidney for over 8 months, living at home, and who was healthy enough to throw out the first pitch at a Red Sox game.
I saw
And
that. That was incredible.
there's a second gentleman who is alive today, about 3 months out, with a pig kidney. So that would have been almost science fiction,
Sure.
only 2 or 3, let alone 5 years ago. And yet we're here. And when it comes to islet cell transplantation, if we look at the progress that's been made on the cells, it's been tremendous. About 2 years ago, we had our first cells approved. Those are the cell-trans deceased donor cells. So they now have an FDA approval. And we're seeing the evolution of stem cell derived cells, and companies like Vertex advancing that technology.
Hmm.
So it feels like we are in a very different point in transplant than we were in the past. And of course, in the therapeutic perspective, there's now us with Tegopubart, and soon when we announce data in November, we should have about or over 100 humans of data post transplantation on Tegopubart. So it feels to me like this is a very different time in medicine and in science when it comes to transplantation, and things that might have felt very far away a few years ago no longer feel that far away. So, do I know the exact timing? I unfortunately don't, in part because we need to see what the FDA is going to tell us they would like to see in terms of our next trial. And obviously, the more patients there are, the longer that trial may take. The FDA also needs to give us some clarity on how long they would like to see data on each patient. So, do they want patients to have 6 months or a year's worth of data before the FDA might consider approving a drug like Tegopubart? So we'll get a lot of that clarity when we have discussions with FDA in the future. But until then, as I just mentioned, we remain focused on islet cell transplantation and hopeful. And our goal is to develop Tegopubart, as quickly as we can, so that we may bring a drug that hopefully will be both safe and efficacious in islet cell transplantation, so that as many patients may benefit as possible.
Wonderful. Can you see those discussions with the FDA potentially beginning in the first quarter of next year, potentially, with
I
these?
think it's early to, it's early to talk about that. We need to get enough patients in, and then we need to get some patient data. Um,
Right.
but um, but I feel comfortable saying we should be there next year. At this stage, it's a little bit early to say specifically which quarter we'll be able to do that. But once we do have the discussions and we get feedback, we will, of course, share that with the community, investors, patients, physicians, and of course, with you.
Absolutely. Yeah, I completely agree with what you're saying. It seems like we've entered a new age. We've turned a corner of some kind. I remember back in 2021, I was sitting in the operating room doing anesthesia when I saw the first update come through that Vertex had a patient 90% off insulin. And I think that was the biggest news to hit the type 1 diabetes community since the discovery of insulin almost 100 years before. And it's just been for the last 4 years. It seems like something new is exciting is happening almost on a weekly, monthly basis sometimes. Um, we certainly are coming into a new age of medical breakthroughs in type 1 diabetes. And I am so excited about Eledon and what you have accomplished so far. Um, One last question in regards to Tegopubart. For somebody with type 1 diabetes, can you speak to what a potential treatment could look like, whether it is an injection, a pill, a subcutaneous shot, and perhaps how often that might have to be taken?
Sure. So today, Tegopubart is taken as an injection. It's an intravenous injection. It starts at the time of the transplant or just before the transplant, and patients receive Tegopubart intravenously every 3 weeks. Our goal, in particular with islet cell transplantation, is to develop a version of Tegopubart that could be injected under the skin, so it would be subcutaneous, to give patients the choice whether they prefer to have a once every 3 weeks or once monthly IV, or inject themselves in the privacy of their home with a subcutaneous version of the drug. We finished work in monkeys and non-human primates on our subcutaneous Tegopubart, but the plan is in the future to move both forward, although starting with the intravenous version of the drug.
Wow, that's incredible. Um, and
But we understand why subcutaneous could be more attractive, particularly for patients who have experienced nor used injecting themselves under the skin. And we'd like to allow patients to have, eventually whichever solution they and their medical provider feel is best for them. Right? Not everyone is going to want to have a subcutaneous injection. Some people prefer knowing that they're seeing a medical professional once a month when they get that IV. There's comfort that may come with that. So if that's what patients choose, then great. Our goal is to have a solution for them. And for other patients that don't want to deal with that and would prefer to have a subcutaneous version, we'd like to have a subcutaneous version of Tegopubart available for them as well.
that's incredible. When could potential human trials begin on the sub-Q version of Tegopubart?
I think let's revisit that next year.
Okay.
As you know, between our data that's upcoming in about 2 months and everything else that's moving on, our focus is to make sure that we can keep the intravenous version moving as quickly as possible.
Right.
But that does not mean that we're in any way that we're ignoring the subcutaneous approach. We're going to advance that as well, but stay tuned for more details on timing.
That's incredible. Yeah, a subcutaneous version could really open up and expand access to an incredible amount of people. It would be like going to the fridge and taking your GLP-1. We got half of America
Exactly.
already.
Exactly.
That's awesome. Thank you so much for your time. I'm sorry that we've taken so much of it, but
No,
you're a busy
it was a
guy, and
pleasure.
we got to let you go. But thank you so much. And on behalf of my daughter and Chris's sons, we thank you for the work you're doing. And we truly believe in Eledon and what you guys are working on. We're so excited for what the future holds. I listen to everything that comes off your website and every fireside chat. I listened to it multiple times. You guys did a great job, by the way, at the last one. I really enjoyed listening to that one.
No, thank you. We, on behalf of all of my colleagues, we appreciate it. And, we don't take the community and the patient support lightly. If it weren't for for people like you and your family, we wouldn't be able to move Tegopubart and what we do forward. So, thank you again for your support, and for your trust.
Thank you so much. Type 1 diabetes, I believe it's one of the most difficult diseases to manage. As a healthcare professional, I've seen them all. And are there worse things that can happen to someone? For sure. But on a day-to-day management, I just haven't found something that is more difficult to manage than type 1 diabetes. My daughter just had surgery this morning, and trying to manage her blood sugar while she's under anesthesia, man, that's a question I get a lot in my group is how do I do that? What should I tell the nurse and the anesthesia provider? And um, it's just such a brutal disease. It's so hard. And we look, we have so much hope for the future. And I do believe that there is a future where my daughter is cured of this disease. And I'm so grateful for you guys and your work.
Thank you very much for having me.
Thank you so much for joining us, DA. We appreciate your time.